Automated systems meticulously sort and prepare these foundational biological components, ensuring each sample is precise.
Within the sterile confines of a laminar flow hood, samples undergo a controlled thawing process, preparing for correctives. The preparation of specialized culture media is paramount, precisely formulated and measured to provide the ideal.
Following thawing, the biological samples undergo a meticulous washing process, removing cryoprotectants and preparing.
Controlled centrifugation at low RPMs allows for the gentle separation of the biological components from the washing medium, concentrating them for optimal use. The result of centrifugation is a concentrated pellet, representing the purified and viable biological material.
A small aliquot of the concentrated sample is taken for rigorous quality control, ensuring the integrity and viability of.
Under high magnification, the motility and morphology of the male gametes are meticulously assessed, a critical step in determining.
Automated cell counting provides precise data on the concentration of viable cells, ensuring optimal ratios for the subsequent fertilization procedures.
While biological samples are processed, the female reproductive system undergoes a carefully monitored ovarian stimulation. The final step in pre-processing involves the meticulous preparation of the oocyte retrieval kit, ensuring
the extracted oocytes are immediately transferred to a precision incubator chamber, maintaining physiological temperatures.
Under precise magnification, individual mature oocytes are carefully isolated from the follicular fluid, ensuring.
Currently, a highly concentrated and motile sperm suspension is prepared, undergoing final selection for the precise intracytoplasmic sperm injection procedure.
A single, morphologically superior sperm cell is meticulously selected and loaded into the micro-injection pipette. The critical moment of fertilization occurs as a single sperm is precisely injected into the oocyte, initiating the
Following fertilization, the newly formed zygotes are immediately transferred to a precision incubator, where controlled temperature and
Approximately 18 to 24 hours post-fertilization check confirms the successful formation of the zygote.
The automated cell culture system undergoes continuous calibration, ensuring the precise environmental parameters and nutrient.
Each developing embryo is meticulously labeled and tracked, ensuring precise identification and continuity throughout the extended culture period.
Advanced time-lapse monitoring systems provide continuous, non-invasive observation of embryonic development, capturations.
The first critical cleavage, transforming the zygote into a two-cell embryo, is meticulously documented, marking a periodically.
Embryos are carefully removed from the incubator for detailed assessment, evaluating their growth and morphological progression. Each embryo undergoes rigorous developmental grading, assessing cellular division patterns and morphology to determine its quality and potential for further growth. By 40 to 48 hours, successful embryos typically reach the 4-cell stage, a vital benchmark.
To maintain optimal growth conditions, embryos may be carefully transferred between specialized incubator chambers.
The culture medium is periodically refreshed, providing essential nutrients and removing metabolic waste products to support continuous embryonic growth.
By approximately 72 hours, healthy embryos typically reach the morula stage, a compact ball of cells.
This bustling main processing area is where the core biological transformations occur.
By day 5 or 6, successful embryos develop into blastocysts, characterized by the formation of an inner cell.
Rigorous morphological assessment under high-power fluorescence microscopy evaluates the blastocyst's structure, symmetry, and
Blastocysts are maintained in extended culture within automated systems, allowing for further development and selection of the most robust and viable embryos. For some embryos, a precise laser-assisted hatching technique is employed, creating a small opening in the outer shell to facilitate successful implantation.
For genetic screening, a specialized biopsy pipette is meticulously prepared, ensuring its readiness for the precise.
With Rizriakiel, a microbiopsy carefully extracts a few trophectoderm cells from the blastocyst, providing material for
Distortions. The minute biopsy sample is meticulously transferred into a PCR tube, preparing it for the amplification of adjustments.
Within the automated PCR thermocycler, The extracted DNA undergoes amplification, generating sufficient material.
That's now then. The amplified DNA undergoes high-throughput sequencing, meticulously mapping the genetic code to identify chromosomal aberrations.
Genetic results are meticulously reviewed by expert bioinformaticians, identifying viable embryos free.
Based on comprehensive morphological and genetic evaluations, the most viable and robust blastocysts are carefully selected for potential transfer.
Based on comprehensive morphological and genetic evaluations, the most viable and robust blastocysts are carefully selected for potential transfer.
Selected viable blastocysts, not immediately used, are carefully loaded into specialized cryopreservation straws. Beds. The automated vitrification system rapidly cools the embryos to ultra-low temperatures, preserving their viability for years. Cryopreserved embryos are then carefully transferred into a liquid non-extoric storage devoir.
This purification and refinement hub represents the pinnacle of precision, where advanced technologies and experts. A comprehensive final embryo grading report consolidates all morphological and genetic data, providing a definitive
richness. When ready for transfer, cryopreserved embryos undergo a controlled thawing process, carefully transitioning them from.
Following thawing, each embryo undergoes a rigorous post-thaw assessment to confirm its viability and readiness for the final transfer procedure.
The selected viable blastocyst is meticulously loaded into a specialized embryo transfer catheter, suspended in
a sterile handover. The loaded embryo transfer catheter is passed to the clinical team, marking the final preparation for the delicate embryo transfer procedure. The final phase focuses on the delicate delivery of the meticulously prepared embryo, guided by advanced imaging. The embryo-loaded transfer catheter is gently and precisely guided into the uterine cavity, ensuring a smooth and atraumatic placement for the embryo.
Under real-time ultrasound guidance, the viable blastocyst is meticulously released into the optimal position within the uterine cavity.
Following the successful release, the transfer catheter is gently withdrawn, completing the delicate procedure and leaving the embryo undisturbed.
A post-transfer verification check confirms that the embryo has been successfully delivered, ensuring no retention within the transfer catheter.
A comprehensive procedure completion report meticulously documents all aspects of the embryo transfer, ensuring thorough record-keeping for future reference.
With the embryo successfully transferred, the focus shifts to the critical post-transfer monitoring phase, awaiting the natural process of implantation.
Within 7 to 10 days post-transfer, successful implantation of the embryo into the uterine wall.
From meticulous scientific precision to the profound miracle of life, IVF represents an From meticulous scientific precision to the profound miracle of life, IVF represents an extraordinary journey.